Okay, good morning everyone. Good morning, good morning. Why others are not joined yet? Good morning everyone. I think Shivani, you missed some classes. Please kindly go through the recorded. Good morning everyone. Good morning. Okay, so friends, in the yesterday class, we were discussing about the complement system and we have finished almost classical and alternative pathway. Is it? Good morning, good morning. Let's finish the little left part of the complement system and we will move on to the T cell development and T cell activation followed by the maturation. I hope you remember, friends, whether it is the classical pathway of complement system or good, whether, oh, please take rest, Shivani, no problem, no problem. Take a complete rest. Yeah. So whether it is the classical pathway or friends, whether it is the alternate pathway, what did I tell you, friends? The major complement molecule, good morning, good morning. The major complement molecule is what, friends? C3. And I also told you to remember, if you want to form membrane attack complex, compulsory, what do we require? We require C5b6 7 8 9 complex. 9 is compulsory. By using C5b6 7 8 also we can make the membrane attack complex, friends, but which is which can give rise to very small hole up to 10 angstrom only. But when we add the poly C9, what happened, friends? We can make the hole on the membrane from 70 to 100 angstrom. Kindly do not get confused with this particular pathway at all. Then friends, what will be the next pathway? Next pathway we will be calling it as, friends, alternative pathway. Everyone listen my words very carefully. Alternative pathway does not need for antigen antibody complex for initiation. We discussed it very clearly. And friends, everyone listen. Alternative pathway is starting from what? Complement 3 molecule. And we have read very clearly in the yesterday class. How does this particular complement 3 molecule is getting cleaved? Does any other protein or any other molecule is involved in cleaving of this particular complement 3 molecule? No, not at all. Complement 3 molecule will be involved in the process of spontaneous hydrolysis. Is it, friends? The complement 3 undergo the spontaneous hydrolysis and it is going to give rise to the complement 3A and 3B. Later we have clearly discussed complement 3B associated with the factor B. Factor B can be cleaved by the factor D. We discussed it very clearly. Now once the complement 3B be formed, it can act like what? C3 convertase enzyme. I told you now, friends, the complement 3B can be stabilized with the help of a properidine molecule. We discussed it. Now this, which is acting like a C3 convertase, break down the C3 into C3A and C3B. There is a addition of the C3B here, and this entire complex can act like a C5 convertase. Now friends, the C5 converters break down into C5A and C5B. Finally, there is a activation of C6, C7, C8, C9 and ultimately there is a formation of membrane attack complex. This is what we have discussed in the yesterday class. Now, there is only one pathway is left, friends, which we called it as the lectin pathway. Kindly don't get confused with this. Why I am using the word lectin pathway? The answer is very simple. We are calling it as the lectin pathway, friends, because here certain glycoproteins are present. What kind of glycoproteins are present, friends? Especially mannose binding glycoproteins are present. Mannose binding lectin is involved in this particular pathway, which that's why we call it as the lectin pathway. It's very easy, friends. In this lectin pathway, only 3 major proteins involved. The first major protein is, friends, MBL, mannose binding lectin. Then the second and third major proteins, which is always associated with what? Which is always associated with this particular mannose binding lectin, which we can call mannose associated serine protease 1 and, friends, mannose associated serine protease 2. These two are the major proteins which will be associated with this particular mannose binding lectin. Kindly do not get confused with this particular point. Now listen, friends, don't get confused. Just listen to me very carefully, friends. Okay, with the red color, whatever I am drawing is the pathogen. Say an example, it is the bacteria. Everyone listen, say it is an example, bacteria. And we all people know, right? The bacterial cell wall is made up of a lot of sugars. Yes or no? Of course, yes. Bacterial cell wall is made up of what, friends? Peptidoglycans. What are peptidoglycans? Which are the large sugars. Correct na, friends? Everyone listen. See, to this bacterial cell wall, abundant amount of sugar moieties are present on the bacterial cell wall. Listen to me, friends. To that sugar moieties present on the pathogen cell wall, who will come and attach us? Mannose binding lectin will come and attach. Because, friends, on the bacterial cell wall, there are a lot of lectin molecules are there, which are sugars. Many lectin molecules are there. To that lectin molecules only, friends, this particular mannose binding lectin molecule will come and attaches. The moment it attaches to the mannose binding lectin, 2 important proteins will be associated. Mannose serine proteases 1 and mannose serine proteases 2. Now listen. This is called as entire complex. Now this complex will start cleaving the C4 complement molecule as well as the C2 complement molecule to initiate the cascade pathway. Do not get confused with that. Friends, the C4 can give rise to C4A, C4B, and, friends, C2 can give rise to C2A as well as the C2B. And because we all know, we people know very well, in the complement 2 molecule there is an exception. Only the complement 2A comes into the picture. In the C4 generally C4B will come. Now the C4B2A it can act like a C3 convertase. Then the steps are common. Now C3 convertase, then followed by the C5, then followed by the 6, 7, 8, 9. Ultimately, friends, there is a formation of the MAC complex. So, friends, all the students listen. One point is very common, whether it is the classical complement pathway, whether it is the alternate pathway, whether it is the lectin mediated pathway, doesn't matter. Ultimate goal of the complement system is, friends, activation of the MAC complex. There is no doubt about it. But the way they activate or the way they form the MAC complex is entirely different. They may approach different ways, but ultimately they will follow the they will form the MAC complex. Because in the classical pathway, if we want to form the MAC complex, undoubtedly we require the presence of the antibody. Without antibody, nothing is going to happen. And in case of alternative pathway, whether antibody is present or not, what is happening? The complement system undergoing the spontaneous hydrolysis, and they will start the complement pathway, and they are forming the membrane attack complex. Similarly, in the I'm sorry. Similarly, in the lectin pathway also we are forming the MAC complex, but with the utilization of the mannose binding lectin and with the help of its associated proteins. In every pathway, formation of MAC complex is the ultimate goal of complement system. And friends, the most important one, whatever the three complement systems that we have studied so far, we started studying about only the B part, correct na? What is happening to the complement B only we understand. It doesn't mean that complement 3A, complement 5A, they are not functional. Friends, they are also having a unique function which you have to remember compulsory for the exam. Friends, in case of matching, you can expect in the part B also, friends. Even you can expect in the part C also. Whatever I am marking here, I want you people to remember at least whichever I marked. So the first one, which complement molecule or which complement complex is involved in the complete lysis of the cell? So which we can call, friends, C5b29. C5b29 is the one which is required for the MAC complex. Friends, if in the exam, if they give C5b28 and C5b29 and if they ask you which required for the membrane attack complex, you have to remember 9 is compulsory required. If only till 8 is present, there will be no formation of the MAC complex at all. Kindly do not get confused with this particular point at all. So this is the first point I want you to remember. Second one, you know very well, suppose example, this is the mast cell or basophil. Inside the mast cells and basophil, we all people know that abundant amount of granules are present. And we also know, in the yesterday class also I told you, on the on the mast cell surface what is present? FC receptors are present. On the basophils what is present? FC receptors are present. On the eosinophils also FC receptors are present. What is the point you have to remember? To the mast cell and basophil, C3A, C4A, C5A come and attaches. And friends, they will be helpful in the degranulation or breaking down that particular granules and releasing all the chemical mediators which are present in the mast cells and basophil. Kindly do not get confused with that particular point. Next one, friends, degranulation of eosinophils. Friends, same. In order to break down the eosinophils, we will be requiring complement 3A and complement 5A molecule compulsory. And remember, all the students understand. Suppose this is the blood circulation. Correct na? Friends, understand what I am talking. These are all the leukocytes. And this is the area where the damage occur, and this is the area inflammation has to occur. Now you all know, right? These all leukocytes has to undergo which process, friends? I told you very clearly. These leukocytes undergo the process called as extravasation. Is it? So friends, they will undergo the process of extravasation and they can enter into the inflammatory site. And friends, the point is very clear. If this particular leukocytes wants to enter into the inflammation site, or if they want to undergo the extravasation, to this FC receptor, complement 3A, 5A, 5B, 6, 7 complexes will attach us and they will help that particular leukocytes to undergo the process of extravasation. You should remember all this without fail. Then the fifth one, aggregation of platelets. I hope you all people remember, even we read in the blood clotting also. First, what will happen, friends? All the platelets come close together, is it? Which we can call platelet aggregation. After that only fibrin threads will be formed. This is what exactly we will be calling it as the clot. Now the question is here, how one platelet come very close or very near to the another platelet? How is that possible? So friends, this aggregation of platelets is also possible with the help of a C3A and C5A. So this 5 you people has to remember compulsory. Cell lysis, degranulation of mast cell and basophil, degranulation of eosinophil, extravasation of leukocytes, aggregation of the platelets. Kindly do not get confused with this particular point at all. Next one, friends, all the students listen. So next one, you remember viral neutralization. Sixth one. What is viral neutralization? If a particular cell, listen everyone carefully, if a particular cell get infected by the virus, now what happened? The complement system will form the MAC complex and they will drill the hole. They will be releasing the perforins and granzymes. I will tell you in the T cells. They will be releasing the perforin and granzymes and they will destroy that particular viral infected cell, which we can call the process of viral neutralization. And friends, this viral neutralization can be mediated by the C5b29, which is the macro complex. But we also discussed among all the complement molecules, C3b is the most pivotal complement, because friends, if this entire MAC complex will be formed, it is compulsory, friends. There will be a requirement of the complement 3b. There is no doubt about it. Is it? So it has to be formed compulsory. Do not get confused with that particular point. Okay. So that's the simple biological effect of the complements, which you have to remember, compulsory for the exam. Please kindly do not get confused and do not forget. Okay, you have to remember this. Okay, so friends, that ends the complement system. Okay, so what will be the next topic that we have to study? T cell. So friends, let's quickly understand what we have studied so far. We have studied cells of immune system. We have understand antigen and antibody interactions. We read what is immunogen, what is antigen. We read about the different type of receptors are associated with the or in the immune system, that is toll-like receptor and soluble receptor. And we studied different type of antibodies also. Correct, na? And we studied the MHC molecule. Good morning, good morning. And we have studied the complement system. Now the most important, where you can get, what I can say, where you can get experimental based part C questions in the T cell also, friends, as well as in the B cell also, compulsory. Very important concept it is, and a kind of tough concept if you really don't understand. But if you understand and if you revise multiple times, the concept will become very easy. Okay, now the question is here, which cells are involved in the production of antibody? Undoubtedly B cell. We know very well. B cells are the one which is divided into plasma cells and memory cells. We know it very well. Now my question is, why we are not studying B cell first and why we are studying the T cells first? Because friends, everyone pay attention. If the B cell wants to be differentiated into the plasma cell and memory cell, first the B cell has to be activated compulsory. Now the point is here, friends, that entire B cell activation depends on the T cell. The entire B cell activation is depending on the T cell. That is the reason, friends. Always remember that. First we have to read how the T cell is maturing, how the T cell is activating, and most importantly, how the T cell is getting differentiated into T helpers, T cytotoxic, and T suppressor, and how they are showing their mechanism of action. Along with the T lymphocyte, we should also read another type of a lymphocyte which we can call the natural killer cell and how it is showing their mechanism. You know friends, listen my words. However, we are studying the maturation, activation, differentiation of the T cell. Similarly friends, we are going to read the B cell also. There is no difference in the process. The process will be almost similar, but the components involved is entirely different. So now let's talk about it. Don't get confused friends. So first listen to me for 20 minutes continuously without interruption. You will get the concept very clearly. So whatever written in the first two slides, I will be explaining very elaboratively in the diagram itself. Pay attention towards this. And we people studied very clearly in the first class, that is cells of immune system, the T cell production occur in the bone marrow. There is no doubt about it, but we studied very clearly friends that T cell maturation is actually taking place what friends in the thymus. Kindly do not get confused with this particular point at all. Now listen. I'm sorry, listen to me very carefully. Hematopoietic stem cell, we read in the cells of immune system, right? Hematopoietic stem cell or the bone marrow, it is going to give rise to what friends? Lymphoid progenitor, myeloid progenitor, erythroid progenitor. We have studied it very clearly. Now this hematopoietic stem cell or bone marrow giving rise to the lymphoid precursor. Every student listen my words very carefully. This lymphoid precursor, it will migrate into the blood. From the blood, it will enter into the thymus. Listen everyone. And it is going to give rise to the T cell precursors. Now friends, this T cell precursor is going to modify it into different types. I will tell you. First, T cell precursor give rise to pro T cell. We are calling double negative. I will tell you what is double negative and double positive. Later, pro T cell converted into pre T cell, still it is double negative only. Once again, pre T cell converted into pro T cell, which we can call it as a double positive. I want you people to remember, when I use a word double negative, when I use a word double positive, we use the word double negative friends whenever the two important surface marker, that is CD4 and CD8, are absent. When these two are absent, then only we can call that particular T cell is the double negative. It's a kind of confusing concept. Pay attention, utmost attention. When we can call double positive friends, whenever there is a presence of the CD4 and whenever there is a presence of the CD8. Kindly do not get confused with this particular point. I can tell you one more line. If the T cell is ready to activate, I can tell you very clearly that T cell contains the CD4 and CD8 compulsory. Anyway, now listen. It is very important for the exam to remember the types of surface markers. Compulsory we have to remember the types of surface markers present at different stages. Now let's talk about pro T cell, which is the double negative, correct na? And friends, please pay attention. What are the surface markers or the surface receptor that are present on the pro T cell in the double negative condition? Why I call double negative? Keep it in your mind. CD4 and CD8 are absent. What are the examples? c-KIT, CD44 and CD25. Kindly do not get confused with this particular point, friends. c-KIT, CD44 and CD25 are the three major surface markers which are present on the pro T cell. Friends, everyone listen my statement. When the pro T cell converted into pre T cell, what are the various surface markers that are getting expressed? Just listen my statements carefully. I'll re-explain. When the pro T cell double negative converted into pre T cell double negative, there will be an inactivation of c-KIT, there will be an inactivation of CD44. Then what is present in the pre T cell? There, listen my friends, there is a presence of CD25 and there will be an expression of CD3, and the most important ones, T cell receptor alpha and T cell receptor beta. These are the very important type of the surface markers that are getting expressed in the pre-T cell double negative. Kindly do not get confused with that. And friends, finally the pre-T cell is going to be converted into what, friends? Pro-T cell, which we generally called it as what, friends? Double positive. When I am calling it as the double positive, and what kind of surface markers are present on the pro-T cell? Keep it in your mind. Pro-T cell contains CD3, pro-T cell contains TCR alpha and beta. You can see very clearly. CD3 is there, alpha is there, beta is there, and the most importantly, CD4 or CD8 is present. That's why finally we can call pro-T cell as the double positive cell. And friends, finally they will get a differentiate, and this CD4 and CD8 surface markers will express it differently. And we know very well CD4 surface markers will be expressed on the T helper cell, we know it. And friends, we also know that CD8 surface markers are going to express it on the cytotoxic T cell. Kindly do not forget, it's your responsibility to remember every single surface marker present on every single stage. It's very important for the exam. Okay, listen quickly. The pro-T cell, which we can call double negative, it consists of a c-kit CD44, CD25. When the pro-T cell double negative converted into pre-T cell double negative, there is a no expression of c-kit and CD44, and the CD25 will be continued. There is an expression of CD3, majorly TCR alpha and beta. T cell receptor alpha beta. Friends, TCR alpha beta are extremely important receptor. Listen my words, which are helpful in contact with the MHC molecule present on the antigen presenting cells like macrophages, like dendritic cell. If a T cell wants to be associated with the macrophage or dendritic cell, compulsory there is a requirement of the alpha or beta chains. Do not get confused. Now friends, finally when we are calling pro-T cell double negative to the pro-T cell double positive, undoubtedly CD3 is present. Anyway, there is a no expression of CD25 also. What we can say, CD3, alpha and beta, CD4 and 8. Then only we can call the pro-T cell double positive. Now, we understand very clearly what kind of surface markers that are getting expressed. Now the question comes here. Friends, we have discussed, we're gonna discuss also. When we are talking about B cell, we read that B cell produced in the bone marrow, and we have discussed very clearly that maturation is also happens in the bone marrow only. But the reason why we are calling only the T cells are getting matured in the thymus, what will be the reason? The reason behind that, in the thymus, friends listen, the T cells are undergoing this selection process, which we call, my friends, thymic selection. Every student listen my words very carefully. There are two type of selections are there. I want you to listen these selections very carefully. It's so confusing. Listen and pay attention. Positive selection and negative selection. You have to listen to me very carefully. And do you think any one selection has to be qualified by the T cells compulsory? No. The two type of selections has to be qualified by the T cell compulsory. If it qualifies only one selection and if it fail in the other selection, friends, T cell can be easily destructed by the thymus. So it is compulsory that T cell has to pass positive selection also and negative selection also. Let me tell you what is positive and what is negative in a simple way. Just listen. Imagine what I am talking. I am a T cell. Just imagine. I am a T cell. Now in the positive selection, what happened? I told you very clearly, every student look into this. On the membrane, what kind of receptor is there? TCR, T cell receptor alpha or beta. I told you, right? In the positive selection, I am a T cell. I am allowed to recognize the self-MHC. What friends? I am allowed to recognize the self-MHC. Whenever I have allowed to recognize the self-MHC, I have to recognize the self-MHC compulsory. There is no doubt about it. So see here, all the students look into the diagram very carefully without confusion, friends. Whatever I am drawing, you can see here, this is the TCR receptor associated with the MHC molecule present on the antigen presenting cell. So I am a T cell. When I was allowed to interact with the self-MHC, I have to interact with the self-MHC. Whenever I started interacting with the self-MHC, I have qualified the positive selection. In case if I am not interacting with this particular T cells, see here, I have failed the positive selection and I will undergo the death, that is the apoptosis. Every T cell is not undergoing the apoptosis, don't get confused. Death by apoptosis of the cells that do not interact with the MHC molecules. Kindly do not get confused with this particular point. All we are not doing, friends, only the T cell which are not associating with the self-MHC. This is what, friends, positive selection. I'm sorry. Now the most important one is, friends, negative. Listen to me very carefully. In the positive in the positive selection, what we have done, listen to me. In the positive, I am a T cell, right? I am a T cell. In the positive selection, I was allowed just to interact self-MHC. That's all. Friends, what I am saying is, whenever a T cell interacted with the self-MHC, it should not interact for the longer time. What do you mean by that? Which means our T cells should not show higher affinity towards the self-MHC. Once again, listen. Our T cells should not show higher affinity towards the self-MHC molecule. If it starts showing higher affinity towards the self-MHC molecule, there is a chance our T cell may destruct the self-MHC. That's why if any kind of a T cell that is showing the higher affinity towards the self-MHC, that can be immediately destructed in the negative selection. Don't confuse, friends. Negative selection that eliminates the thymocytes or the T cells which are bearing high affinity receptors for the self-MHC molecule. So only that kind of T cells only getting destructed. You can see here, if in case of a negative, see, this is the positive selection. What we have done in the positive selection, I hope everyone remember. In the positive selection, we just checked. What we checked, just whether our T cell receptor are associating with the MHC molecule or not. It has to be associated compulsory, but it should not have high affinity. In the positive selection, we will check whether it is having the affinity or not. We are checking only the affinity. It can attach or not. If it is unable to attach, we will destruct. In the negative selection, we will check whether it is having the minimal affinity or the higher affinity. If it is showing the higher affinity, friends, what we are going to do, we are going to destruct that particular T cells compulsory, whichever showing the higher affinity towards the MHC molecule present on the macrophage and dendritic cells. If it is not showing higher affinity, if it is showing a minimal affinity, what will happen? Now T cell never die, and ultimately, friends, the T cells are getting differentiated to give rise to T helper cell. And friends, T cytotoxic cell, and we all people know very well T helper cell on its surface it is having CD4 and cytotoxic cell on its surface having the CD8. This is the positive selection and this is the negative selection. Friends, I want all of you has to remember a point without fail that it's not only T cell. Listen my words carefully. It's not only T cell, even in the B cell also. Even in the B cell also, there should be a compulsory signaling. There should be compulsory signaling process. The signaling process never be asked in the exam. But friends, in order to understand the T helper cell activation, maturation, differentiation, it is very compulsory. We have to understand the signaling. We have to just understand. You no need to remember for the exam point of view at all. Pay attention. How does the signaling occur? Just to understand enough. Okay. All of you listen to me very carefully, friends. You are able to see. This is what cell? This is the membrane of antigen presenting cell. Now, friends, this particular antigen presenting cell having MHC, on the MHC it expresses the antigen. We know that antigenic peptide. Now you can see, right, this is what, friends, this is the T cell membrane. So on the T cell membrane what are present? Various type of T cell receptors are present. What are the major receptor? I have told you alpha, beta are the major T cell receptor. Every student has to look into the diagram very carefully. Now MHC molecule and TCR alpha or beta associated with each other. Is it or not? Yes, they will be associated with each other. Now listen, along with that, friends, it is also associated with the CD4 or CD8 depending on the type of the T cell. Now what is the point I want you to know is, along with the alpha and beta receptor, there are many co-receptors of T cell is there. They are also getting activated. The question is very clear. What is the question? When this particular, listen my words very carefully, when this particular co-receptors are getting activated, friends, co-receptors are getting activated whenever MHC, TCR alpha, beta and CD4, 4 or 8, when all these get activated, then only there is an activation of co-receptors of the T cell. What are the various co-receptors? Epsilon, delta, gamma, zeta. What, friends? Epsilon, delta, gamma, zeta. Alpha, beta are the major receptor. These are all the co-receptors. Just understand what I am talking, friends. When all these co-receptors get activated, you are all people can see, right? All these co-receptors are having what kind of domains? Cytosolic domains. With what kind of activity, friends? They are having cytosolic domains with the intrinsic kinase activity. Kindly do not get confused with that particular point. So what's happening, friends? Ultimately, the phosphorylation occur and there will be a activation of the different type of kinases. I don't encourage you people to remember all the type of kinases which are getting phosphorylated, which is not at all required for the exam point of view. Just listen. So these are all the zeta, gamma, delta, epsilon. Once all they get activated, they will undergo the process of phosphorylation. You can see here they are going to activate a different type of kinases. These are all series of kinases that are getting activated. You no need to remember. What I want you to finally remember is, everyone listen, what I want you to remember. Don't get confused. Upon continuous activation of different kinase molecules, finally they activate an important effector molecule. What do we call it, that important effector molecule? We will be calling it as, friends, PLC gamma, which is an important effector molecule. Friends, this particular PLC gamma, what it is going to do? It is going to break down the PIP2, which is a membrane bound lipid, we know it. Listen my words, listen my words. All this series of kinases activating the PLC gamma. PLC gamma break down the membrane lipid PIP2 into IP3 and DAG. And everyone commonly know that, friends, IP3 will go and bring the calcium. Now the final point, that DAG and calcium together will be activating an important protein called as a PKC. Now everyone listen. Friends, why we have done all this? Now this PKC ultimately is going to activate many genes. Listen my statements very carefully. This particular PKC is going to activate many genes. What kind of genes can be activated by the PKC? The genes which are involved in the T cell development. This is why the process of signaling has occurred. Such a simple step it is. Don't confuse and don't make it complicated. You have to understand one point very clearly. Why does the process of signaling is occurring? Listen my words. Why does the process of signaling is occurring in the T cell development? The major aim of the signaling process is to stimulate the genes which are involved in the development of T cell. So how that particular genes can be stimulated? By a protein, by a kinase called as a PKC. How does this particular PKC is getting activated? Now when the TCR alpha and beta associated with the MHC, there is an activation of several co-receptor, and the co-receptors are having intrinsic kinase activity, and they are all get activated which are a series of kinases. Finally they are activating an effector molecule called as PLC gamma, and PLC gamma break down the PIP2 into IP3 as well as the DAG molecule. Now IP3 is going to give rise to what, friends? Calcium. Now DAG and calcium come close together and stimulate the PKC. Then PKC will be helpful in the development of the many genes which are involved in the T cell development. Simple it is. That is the signaling process. Okay. Now friends, these two slides are extremely important slides. Just listen. One point is very clear. T helper cell, not the T helper cell. You can directly use a word T cell. T cell can show autocrine signaling. T cell can show the autocrine signaling. And another important point I want you to remember, friends, T helper cell, it plays a key role in the activation of cytotoxic T cell also. At the same time, T helper cell plays a very crucial role even in the activation of the B cell also compulsory. So that's why I can tell you, when compared to the cytotoxic T cell, T helper cell is the most important one, because you can't imagine the functioning of a cytotoxic and B cell without T helper cell. And all the students listen. How the T helper cell is getting activated and what is the pathways involved in their activation. All the students look into the diagrams very carefully. This is normal antigen presenting cell. What is that? Macrophage or dendritic cell. Don't get confused with this particular point. And friends, this is what cell, friends? This is the T cell, right? Now listen to me very carefully. You can understand the picture. This is what, my friends? MHC molecule, there is no doubt about it. And this is what, friends? T cell receptor, there is no doubt about it. And in the middle, light blue color, whatever I am drawing now, the red one, either CD4 or CD8, just imagine, which co-receptor. There is another co-receptor. You can ask me, sir, what is the major receptor? I can tell you MHC and TCR is the major receptor, and these are all co-receptors. On the T cell, friends, pay attention. On the T cell, there is a CD28 receptor. You have to remember this. and the CD28 receptor present on the T cell associated with the B7 receptor present on the macrophage, which is acting as a co-receptor. Now listen, whenever there is an interaction of the major receptor and co-receptor, who is getting activated? T cell is getting activated. Once the T cell get activated, inside the T cell there is an important gene is present. What, my friends? Inside the T cell there is an important gene is present, which we can call interleukin 2 gene. Now that interleukin 2 gene get activated and it releases various kind of interleukin molecules. Interleukin 2. The point is very clear. Interleukin 2 molecule is released by the T cell. There is no doubt about it. Now friends, where does that interleukin 2 molecule will come and attach us? They will come and attach us interleukin 2 receptor present on the same cell. Kindly do not get confused here. That is the reason we will be calling it as the autocrine signaling. Now you can ask me a question, valid question. Sir, what is the use of this interleukin 2 molecule? Interleukin 2 molecule will help the T cell to undergo the several divisions. After the division only, right friends, the T cell is undergoing the process of differentiation. Kindly do not get confused with this particular point. If the T cell wants to be divided, there should be a requirement of compulsory important cytokine. See, interleukins are cytokines only, right? The example of an important cytokine is the interleukin 2. Who is releasing the interleukin 2? T cell only releasing, and it is attaching to which cell? T cell only. Don't get confused. Look into this, look into this. Very important slide. Macrophage, T cell, and what it is releasing? Interleukin 2. Friends, all the students listen my statements very carefully. Whenever macrophage and T cells associated with each other, T cell is getting activated at the same time. Macrophage or dendritic cell is also getting activated. Don't get confused. Listen my statements very carefully. The activated macrophage or dendritic cell will release interleukin 12. And we just now studied that T cell is going to release the interleukin 2. Now friends, this interleukin 2 and interleukin 12 will go and activate the T helper cell 1. Now you know what the T helper cell 1 in return it is doing? It releases the interleukin 2, IFN gamma, TFN beta. These questions are also asked many times in the part C and part B. Please remember. T helper 1 releasing IFN gamma, interleukin 2, TNF beta. What is the reason? They are all responsible for the hypersensitivity reaction. They are all responsible for complement fixation. You have to remember that compulsory. Which are activating the T helper 1? Majorly interleukin 2 and interleukin 12. Friends, listen. Whatever the interleukin 2 released from this T cell, it will come and activate the T helper 2 also. But if they ask you a question in the exam, which type of interleukins are majorly plays a key role in the activation of T helper cell 2? Always remember interleukin 4. Interleukin 4 is the major activator of T helper cell 2, whereas interleukin 12 and 2 are the major activators of T helper 1. Please don't confuse. Now this interleukin 4 activate the T helper cell 2. T helper cell 2 in return, it releases interleukin 4, 5, 6, 10, 13, 16 and 17. These are all the different type of interleukin molecules which are released from the T helper cell 2, which are responsible majorly for the activation of different type of antibodies and activation of the eosinophilic molecules. Kindly do not get confused with this particular point. And you have to remember a point. If the T helper cell 2 wants to be activated, there is a requirement of TH1. And this later we will study. The TH2 only go and activate the B cell. How? I told you right just now. Th2 is releasing several interleukin. These interleukins only will activate the B cell. Then only B cell is undergoing the process of differentiation. Till this slide, what we studied is just activation. Now in the next slides, we will understand in the tomorrow class how T helper cell will destroy the targeted cell, how cytotoxic T cell destroy the targeted cell, how natural killer cell destroy the targeted cell elaboratively. Okay. Friends, is it clear everyone? I know it's a kind of rigid concept. You require 2 to 3 times of revision. Then only you can remember for the exam. Okay. Clear everyone? Listening the lesson is not important, friends. You have to read it compulsory. Okay. Clear? I hope it's clear for others. Okay. Friends, we have an immediate class at 8:30 a.m. I'll share the link now. We will be continuing the process of gastrulation. Okay, I'll be sharing the link for the developmental biology class. Please attend the class. We'll finish it off. Okay, friends. Thank you all. Bye-bye.